Thank you very much, Paige. Well, welcome everybody, and thank you, Paige, for setting this up. My name is Antoni Wandycz. I'm the product specialist for SeaHorse. I'm joined today by Anup. I'll be giving the presentation today, but Anup will be joining and helping answer any questions that you may have. And I'll have a couple of pauses during my talk, and then certainly at the end, feel free to ask questions. So we're here for you. Let me turn off my video to make sure that everything goes smoothly. So, you know, over the years, we've really talked to a lot of people about the SeaHorse technology. And those who haven't used SeaHorse but are interested in learning about cell behavior, and especially if you're introducing some kind of perturbation to the cell, folks tell us that the SeaHorse, although it's interesting, it's not, their focus isn't metabolism. They prefer to leave that work to others. Now, it's understandable given the complexity of technologies in the lab today, but what we tell them is that out of the thousands of SeaHorse publications, many were actually published by biologists who are not metabolism experts, or mitochondriacs, as some were called, but were simply interested in the function of the cells. So they soon realized and appreciated that getting a sensitive and real-time readout of the indicators of glycolysis and mitochondrial respiration really added a lot of valuable information to their studies. So today I'll give a brief and quick overview of SeaHorse, and so we welcome your input. In your chat, if you have any questions during the presentation or afterwards, we'd love to know about what you're working on and what made you interested in the SeaHorse. And I want to emphasize that no matter which groups are represented, infectious disease, neuro, basic cell physiology, you know, what SeaHorse XF measures is so foundational to the basic cell function that it can nearly apply to every field in biology. So I'll touch on examples in immunology and cancer, but with so many publications in a variety of disciplines, we can show you how to search for your sample types at the end of our talk today, and know that the examples that we show can be sometimes applicable to your cell types. So, and Anup and I can also follow up with individual labs after today with any additional questions you may have. So if you're planning to do an instrument, maybe a SeaHorse instrument demo in early 2022, I'll also have some important updates regarding product development and new assays, and we can have another joint session just prior to that SeaHorse demo. All right, so I'll focus on SeaHorse today, obviously, but I wanted to make you aware that Agilent has a lot of different technologies that can help you, especially if you're looking at single-guide RNAs and CRISPR. We have a group of called Novocyte, or from ASEA, a Novocyte line of benchtop flow cytometers up to 25 colors. We have a line of cell impedance technology instruments from also ASEA. This line is called Excelligence. And of course, our biotech colleagues, this is a company that Agilent acquired a couple of years ago with their plate readers, washers, and lines of imagers like this Citation Model 5 shown here on the screen. And by the way, this citation can be connected to the SeaHorse for cell normalization. I'll touch on that in one of our slides. Now, as you know, live cells are not static. They're constantly undergoing some kind of change. They're growing, activating, dying. And SeaHorse gives you one of the best ways for you to see and hopefully understand these changes as they happen. And, you know, while endpoint assays are powerful and they're going to be here with us to stay, you can gain an even better understanding of the dynamics and the cell behavior over time by adding functional data like SeaHorse. And if you limit your data to just snapshots in time, you may miss very important events that you will only see in real-time analysis. And more reviewers are asking for, and sometimes even requiring, real-time functional data to be included in a publication. So SeaHorse is one of those ways that you can augment your endpoint assays. So whether you're looking at cell activation, differentiation, understanding mechanism of action and substrate utilization, you can ask questions like, what is driving my cancer proliferation? How do I know when my stem cells have differentiated? When are my immune cells activating? What's causing my cells to die? So because of the nature of the data from SeaHorse assays, you're able to understand the drivers of cell behavior through the lens of metabolic function. Now, many labs around the world are using these analyzers, and the communities are growing quickly, especially in the fields of cancer, immunology, cardiology, and neurobiology. Experts continue to publish high-impact papers with SeaHorse data, and they're using more and more of Agilent's newest applications and assays, a few of which I'll cover today. So with a fast-growing community of users, you're not alone as you start the SeaHorse technology. Aside from the publications, you can lean on our technical team for help. We have a staff of tech support scientists, and Anup on the call with us today, who's a dedicated application scientist who can help with any data analysis that you may have, even if you're looking through some publications and you find something interesting and you want to better understand that data even before you have a SeaHorse instrument on campus. He can guide you through some of this data and explain what those things mean. So we can really personalize the experience of SeaHorse that's relevant to your specific research goals. So let's look at a couple of different types of SeaHorse instruments, or the 3 different types that we have. Currently 3, coming on 4. So today we have the smallest 8-well instrument. It's called the HS Mini, starting from the right here. And then we have the larger 24 96-well version. Now all measure the same thing, but aside from throughput, each type has a unique strength. So the HS Mini, formerly called the XFP, which I think there is one on campus at ASU. This is an 8-well system, but the HS Mini is the HS stands for high sensitivity. It has a special plate that allows you to use fewer cells, sometimes 3 or even 4 times fewer cells per well than the previous generation. Better improved software, better sensitivity overall. And so this instrument is used very commonly in for cell populations that are rare, maybe some precious primary samples that you have, or immune cells that are maybe very quiescent, and you need a lot of them. And so with these kind of instruments, you can sacrifice fewer mice for your experiments. It'll cost you less in the long run. So people have been asking for this, and we finally came out with this about a year ago. The 24-well system is best used for tissue samples, at least certain maybe tissue slices, brain slices, things like that. There's a special islet plate, and so for those that are studying diabetes or anything kind of related using islet cells, this is a perfect instrument for that. There are lots of publications on that. People have used the XFP24 for whole organisms or for very highly energetic cells as well. So by whole organisms, anything that can fit into that well. So maybe C. elegans worms or Drosophila, maybe a part of the Drosophila. There was even a group at UCSD that was attaching a Drosophila, a live beating heart, adhering it to the bottom of the well and testing epinephrine and how that was changing the metabolism of the cells in there. It was pretty incredible. And the XFP96 is kind of our flagship model, most popular model because it has a lot of flexibility. You can run a lot more samples on it. It's great for immune cells. The well sizes are similar or the same actually as the HS Mini, not counting the special HS Mini plate. That's a separate plate. But the XFP96 also has a special spheroid plate that can accommodate organoids or spheroids up to 800 microns in diameter. Now, I mentioned there's a 4th instrument. It's actually kind of an evolution of the XFE96. And so you'll see both of those instruments in January. The XFE96 is going to continue to sell, but there's a newer version, maybe not applicable for as much for academic institutes initially, unless you're a lab that's doing a lot of drug discovery and kind of pharmacy or pharma focused. So it's really tuned for the pharma and commercial customers, but of course it will be offered to everybody. And the difference is that it does have better performance, lower limit of detection, better CVs, improved software features, some other functionality in there, better fit for automation, and et cetera, some more things there. So perhaps when we meet again in early 22, we can bring that up in more detail. So these are real-time kinetic measurements. Typically, assays take about an hour or two, but you can start seeing results within the first few minutes. And you can also extend your assays up to 12 hours if needed, and if the cells are happy. The software is really easy to use, and it really helps you set up your experiments very quickly and get you going. It's available both, the software is available both for desktop, PC-based, unlimited licenses, you don't have to pay for extra licenses. And then there's a cloud version that we released called SeaHorse Analytics that also offers some advantages in addition to being great for Mac users because it's browser-based. Most people run a monolayer of cells, but several labs routinely run tissues, whole organisms, or spheroids on a seahorse. And you don't need any labels to run your assays, just need the sensor cartridges, which detects the two analytes, protons and oxygen. There are 4 injector ports for each well, and many of our assays use a combination of 2 or 3 compounds, leaving you with 1 or 2 ports for your compounds of choice. And lastly, we have fully validated kits and reagents to ensure reproducible data and get you started quickly. So no need to mess with a pH meter or troubleshoot correct formulations. If you know the basics of cell culture and how to use a pipetter, you can definitely run a seahorse assay. The seahorse itself is quite simple to use. And so one more point about the 4 injector ports I just mentioned. You see a close-up of the sensor plate here and 4 ports. I think this rather understated capability is, like I said, is understated. It really allows for limitless combinations of experiments. Nearly every single type of cell or sample types, primary cells, cell lines, bacteria, platelets, organoids, and then you multiply this by the various compounds that you can inject, and then the programmable nature of those injections, and you can see the incredible variety of experiments that you can perform. And so there are so many biological questions that you can ask the seahorse. Your grant proposals and your publications will be that much more compelling. Reviewers put a high value of real-time functional analysis and how it can augment and validate data from your endpoint assays. So this is a really important point that a lot of people appreciate now about seahorse. So let's dive deeper a little bit and see how this works. Some really clever engineering really provides a dynamic readout of what the cells are doing over time. And let's see if the video will play here. So by lowering the sensor into the well, a temporary microchamber is created where the measurements take place every few seconds with 3 minutes on and 3 minutes off. This is programmable, but it's preset with our assays. So the sensor only touches the media, not the cells. And this results in a very fast and highly sensitive readout of how your cells are behaving metabolically in real time. Now, what are we measuring and how does it work? You know, our approach is that our approach to cell metabolism is to measure the rates of these 2 key pathways of energy production. And the premise is that any change in cell function requires a change in energy production. So no matter what the assay, we measure 2 things in parallel: extracellular pH and oxygen consumption. Mitochondrial respiration is important in driving homeostasis, longevity and persistence, where glycolysis is short-lived and generally used for high energy demands, quick responses to infections, and so on. You'll see OCAR, or oxygen consumption rate, and ECAR, or extracellular acidification rate, used in this presentation, and you'll probably may have seen in the past in many publications. And when you start reading about SeaHorse, you'll see these terms over and over again. So when you see OCAR and ECAR, you now know what that is. And then PER is a newer term that's used in a couple of our assays, the ATP rate assay and the glycolytic rate assay. And PER is essentially a quantitative version of ECAR, so quantitative version of the extracellular acidification rate that's used in these two new assays. And we did that because in that new assay, like the ATP rate assay, because the denominator is the same, we have picomoles per minute in both PER and OCAR, you can then compare these two and you'll get a readout, a quantitative readout of the contribution of both of those pathways to the overall ATP rate. So SeaHorse can provide yes or no answers, and you really don't need to be a metabolism expert. Of course, it helps if you know biochemistry and metabolism, but if you want to learn more about those topics and your project involves some of those aspects, SeaHorse is really a fantastic way to do that. And so you can ask basic questions. Are my cells healthy? Is my particular cell type activated? Is my cell population ideal for reprogramming? What is the metabolic signature for the cell phenotype I'm looking into? So some great questions that you can ask of the SeaHorse. Now we have a variety of assays, and you can start with a broad assessment of your cell function or dive much deeper into the mechanistic understanding of what's going on with your cells. I won't go through all the assays, but know that you can start from the top looking at the ATP production assay and then go to the cell mitostress test kit, which is probably the most common, not probably, it actually is the most common assay used, although ATP production rate assay, because it's so simple, just uses 2 injections, oligomycin and then the rotenone annamycin A. Those 2 assays are a fantastic way to start and get familiar with the SeaHorse and cell metabolism. And you can get to, like I said, you can start with a broad assessment of cell function, or you can dive much deeper into specific cell mechanisms, like pinpointing an issue with complex 1 and the electron transport chain, for example. So some of our newest assays, like the oxidation stress tests, are built on the mitostress test by adding an inhibitor to block one of the 3 more popular substrates to help you understand your cell's utilization of those fuels and how that impacts cell function. A quick high-level overview of the actual workflow, and we'll spend more time on this when we get closer to the demo, but essentially it's a 2-day workflow. Day 1 or day prior to the assay is a very simple step. It just takes a few minutes. You need to hydrate the sensor that I showed you with the fluorophore that's embedded on the bottom. That's the sensor that detects the two analytes. So that needs to be hydrated. And then you plate your cells, particularly if they're adherent. If you are working with cells in suspension, you would use a different protocol, but you would just simply use either PDL-coated plates that we offer or coat the cell plates yourself with some kind of adherent, like cell tack from Corning or others. So 2 different protocols, but the day of the assay, it becomes the same thing, where you prepare and load all of the kit reagents that we offer, and then that takes, for a 96-well instrument, takes an hour, maybe a little more, and then you run it. There's a 15-minute calibration prior to each assay. You start the assay that runs from an hour and a half, or actually from an hour to 2 hours, depending on what kind of assays you run and parameters. and then you get your data. And the data you can analyze right on the monitor that's included with the instrument, or you can download a data analysis application on your own PC, or run it on the cloud now with the SeaHorse Analytics. And that can be done later. It can be shared with folks like Anup or tech support or anybody else if you have questions, or share with your colleagues. And I mentioned at the beginning of the talk this biotech citation. This is an imager that pairs nicely with a SeaHorse, and it uses both bright field imaging to do a QC check at the beginning before the SeaHorse assay starts. And then when you're looking at maybe cell counts and you want to use cell count for normalization, which is a very highly recommended way of doing normalization, you would stain your cells and then you'd put it back into the citation instrument at the end of your assay, and that will give you an accurate cell count because it uses like a Hooke's dye to stain the nuclei, and that data is fed and it's controlled all by the SeaHorse software so that you can get the normalization data included. You don't have to manually enter anything. And it also saves the images of your cells with every plate. So you can always do investigation later on. Maybe you're submitting your work to peer-reviewed publication, and then you have to go back and do some QC and add some more data to it. You can always go back because it's always saved there. And then for high throughput experiments, you can always connect it to an automation platform like the one that Agilent offers, the Bravo instrument. Now, of course, consistency and reproducibility is key, and SeaHorse has a very good track record of it. This is one particular example from a biopharma company, actually it's from a large pharma company and GSK on the East Coast, that they were screening over a 6-day period, and they wanted to see the results and how reproducible the SeaHorse is, and this is a good indication of that reproducibility and the robustness of the data. All right, so I'll pause here before I jump into some examples. Are there any questions that you have? You can submit it to chat. Anup, if you can kind of monitor the chat and read the questions, or we can save those questions to the end.
I have a question if you can hear me. Can I just vocalize it?
I can, yeah.
Oh, okay, great. Yeah, so my lab works with organoids. Closest that you mentioned was for spheroids. So I'm wondering if you have any suggestions as how to normalize for cell number, or how you would normalize to get to, keep a consistent baseline.
Yeah, that's a good question. So it's kind of that area where people have asked that question about tissue as well, and you will probably have some comments as well. But my first kind of response to that is, depending if you have consistent sizes of these organoids, you can probably, if you're using one organoid and you know approximately, maybe you know how many cells are in that organoid, maybe you don't, maybe it's by weight or size of some kind. That's one potential. Anup, do you have any experience with organoids and normalizing those for assays?
Yeah, that's a really good question. And Antoni, you're right. Like when we are working with spheroids or organoids in SeaHorse, the way we normalize them is by looking at the size and the number of cells in each spheroid. So you would have a good idea like how many cells are there in each spheroid. And if you keep your protocol consistent, and you can assume that you have the same number of cells per spheroid. And you can normalize either to cell numbers of the spheroid, or you can just normalize to one spheroid if you're using only one in your web.
Oh, so you actually can just use one organoid, one spheroid, that works?
No, we have to optimize.
You could.
Yeah.
Go ahead.
Yes, yeah, you could, because it depends upon like each lab has a different protocol, and then the numbers are dependent on the cell type also. So I mean, we don't know how many we need for intestinal organoids, but maybe we have to look up some reference paper will be there. But otherwise, we have to optimize that in your culture conditions, how many cells are there in each spheroid, and whether one spheroid is enough to give a optimum OCAR or ECAR readings.
Yeah. How large are those organoids, if I may ask? Do you know?
I guess it
In
depends.
diameter?
Sorry, are you asking me or Anup?
Yeah. If you know the diameter of
Oh,
those organoids that you're working with.
I guess it really depends on how long we let them grow, because
right.
you know, a 1-day organoid is much smaller than a 3-day organoid. So they could be anywhere between, I guess, 200 microns in diameter to, you know, much larger than that. Like, I don't know, even get up to 800, I guess. And that was actually another question. You said 800 for the spheroid plate. Did I hear that right? Like the location or the divot. I actually, I'm sorry to take up so much time here. I was just kind of curious about that plate itself. Is it,
Yeah.
I've tried to optimize a little bit with SeaHorse, and I wasn't sure if the spheroid plate, how different it is than the normal 96 well plate.
Yeah, it's quite a bit different, and we have a technical document that shows kind of the specs of that plate, but it's the plastic, the shape of the well, and the size, the volume that then you can, you know, the probe is lowered like the cross section that I showed you, and it creates this temporary microchamber. And so with a spheroid plate, you have a larger chamber which can accommodate these larger samples, which then, because you don't want the detector touching the cells, you still just want it to be touching the media. And then you want enough space in that well so that if the organoid or spheroid is respiring normally, you know, and producing protons and absorbing oxygen, that it's kind of in a normal, happy state. And then it's, as Anup pointed out, if it's energetic enough to produce enough signal for us to test, or for the SeaHorse to detect, then maybe one organoid is enough. But it really depends on the normal kind of energetic state of those cells within the organoid, and whether that can be detected or not. And so, yeah, we can send you information. I can send you later on information about the spheroid plate, and we can have an offline discussion, you know, going into more detail about that.
Fantastic, thank you.
Sure. All right. I don't see any other questions in chat. So, you know, immune cells are highly integrated, relying on a cascade of signaling networks, checkpoints, and things to regulate cell processes. And we have proliferation assays, flow cytometry, and cytokine assays that provide a view of immune cell fate and function after the immune cell has committed to a particular outcome. But if you're doing some kind of pathway intervention and modulation to control immune cell function, you really want an understanding of the upstream drivers of the immune cell processes. So using SeaHorse and understanding cell bioenergetics, a lot more now is understood and known that the cell bioenergetics is known to influence many of these pathways, and is both an indicator and a controller of immune cell fate and function. And there have been a lot of papers recently, and this kind of, our group has come up with this nice picture that summarizes what we've learned, particularly about T cell biology. But it can apply to other types of cells as well. But in the case of T cell biology, we have a deeper appreciation for the dynamic connections between the metabolic poise and immune cell function. And thanks in large part to experts in the field like Erica Pierce, Christoph Hess, Greg Delgoff, and several others. And the bioenergetic equilibrium can be measured by looking at the ratio of OXPHOS over glycolysis. And in immunology research, glycolysis, or ECAR, is used as a biomarker for activation. Glycolysis is highly dynamic but short-lived. And you can see that as the naive T cell goes to an effector T cell state, Greg Delgoff and others have actually shown and published papers that show that glycolysis is actually the requirement, the requisite before the transition from a naive T cell for that T cell to become an effector cell and fight off infection. So very relevant in today's world of more highly kind of more attention paid to infectious disease and infections in general. And so OXPHOS and an increase in mitochondrial function is also critical, and it's important for longevity and homeostasis and fitness. And when you see this life cycle of T cell, the equilibrium is different, and the steady state is what sets them up for a specific fate and to set them up to perform a specific function. And so I'll show you a couple of things here later from a lab that's doing immunotherapy and trying to balance between these two states, having tumor killing and having a cell be very glycolytic and having that tumor killing capacity, but then wanting cells to last in your body for years and years so that it can protect you from future infections. And since the SeaHorse ATP rate assay measures both pathways simultaneously, and you'll probably use this assay during a future demo of SeaHorse, you can easily calculate the total energy production and the contribution that total from each of the two pathways. So like in the previous slide, you look at the energy dynamics over time as the cell function changes. In day 0 or 14, even though the total ATP is similar, the ratio of those two pathways and the contribution is different. And in addition to looking at the ratio of OXPHOS to glycolysis at basal condition, you can take the next step and use the cell mitostress test to look at a variety of other parameters focused on mitochondrial function. So I mentioned Erica Pierce before, and she was really one of the early pioneers who demonstrated using this assay, this mitostress test kit, that something called the SRC, or spare respiratory capacity, was key in identifying and predicting the potency and persistence of immune cells. So often the greater the spare respiratory capacity, the longer a memory T cell can persist. And so let's see if I wanted to, this was the slide I was mentioning. This is from Carl June's lab at UPenn. And so labs focused on immunotherapy, like the June lab, have been using SeaHorse assays, including mitostress tests, to engineer and monitor these engineered CAR-T constructs and fine-tune the function of these cells for optimal killing. So one of these constructs drives the cell to be more glycolytic for better tumor killing, and the other makes it more oxidative, required for persistence. And this is just to show additional data from the same paper and lab where they used the mitostress test to test the quality of the mitochondria in these CAR-Ts with different co-receptors. Now, if you're working maybe with different immune cells like macrophages, labs like the Ajit Devkaruni lab at UCLA are able to phenotype macrophages based on the metabolic activity. And macrophages are impressive. They have a lot of different functions in the body, and at times they seem to be opposing. M2 macrophages are anti-inflammatory and are a bit more oxidative in nature, whereas the M1s are pro-inflammatory and mostly glycolytic. If you aren't sure what type of macrophages you have, you can run a quick SeaHorse ATP rate assay to metabolically phenotype them. Now, switching over to cancer, you've probably heard the Warburg effect, when cancer cells preferentially use glycolysis, and then maybe the Crabtree effects, which is like a glucose-induced suppression of respiration. And these are both metabolic signatures of cancer growth. With SeaHorse OCAR and ECAR readouts, you see the total ATP production rate in cells and the fraction coming from OXPHOS and glycolysis. So using a representative panel of frequently used cancer cell lines, we found that even in cells typically considered to be highly glycolytic, the ATP production from glycolysis is only between 30 and 60% of the total ATP production. So this assay can be used as a great way to quickly screen or phenotype your cancer cells. Another example is from Wayne State, and you can use SeaHorse to get a quick assessment of the chemo resistance of your cells. In this particular example, they tested ovarian cancer cell lines and found increased ROS production, mitochondrial mass, preference for palmitate, and a decreased sensitivity to glucose deprivation. ECAR here is glycolysis, as a reminder. Using a SeaHorse phenotype screen, they showed that both cell types were glycolytic, but that the chemo-resistant cells were more oxidative. So again, a great way to get some quick answers. This is a really cool example from Roswell Park Cancer Center in Buffalo, New York. This lab was particularly interested in exosomes secreted by melanoma cells. So the tumor stroma around these melanoma cells, especially the fibroblasts, are being reprogrammed by these exosomes. They ran a mitochondrial stress test, and now you're looking on the top left-hand side, you're looking at OCAR over time. When they expose these fibroblasts to increased concentration of exosomes, they see a decrease in mitochondrial function. Now, the question they asked, and you may be asking, is are these cells dying, or are they switching to glycolysis? So we see in the glycolytic rate assay, the top middle right column, that the glycolysis is increasing at the same concentration that exosomes in the mitochondrial stress test have a decreased oxidative phenotype. So another glycolytic rate assay was performed, that's the bottom chart, with inhibitors to these two microRNAs, confirming that the glycolytic switch does not happen with these two, without the two microRNAs. And this picture illustrates what they believe is going on. So these melanoma cells release the exosomes containing these two microRNA culprits. They're taken up by the surrounding fibroblasts. Those metabolically reprogrammed cells then migrate to a non-tumor and more oxidative environment and transfect the normal stromal fibroblasts, making them more glycolytic. These events are basically lead to a more acidic and tumor-friendly environment, making it much easier for melanoma cells to invade and expand. So this example highlights three things that are quite important. That understanding the pre-metastatic niches need that, understanding that the pre-metastatic niches need an acidic environment, so you can look for glycolysis in tissue stroma for evidence of new metastatic sites. You can look at the biomarker activity of glycolytic proton efflux rate, or PER, with some anti-microRNA therapy. And then you can quickly check whether the microRNA inhibitors are actually working. And then third, you can look at the glycolytic levels in fibroblasts. If normal fibroblasts have been transfected with exosomes containing the two mirrors, it may indicate pre-metastatic priming. So here's another glimpse at the chart that I showed you earlier. Again, these assays are just the beginning. The SeaHorse technology is very flexible, and you can start by asking very complex questions, especially as you get more advanced in your experimental design. And so some of these questions, so I talked about the ATP production assay, talked about the cell mitostress, the glycolytic rate assay as well. And then there's several others, especially if you're working with long-chain fatty acids. Or if you're thinking about working with isolated mitochondria, there's a product, if you look on the bottom left of the screen, that is cell permeabilizer. So it allows you to use this recombinant protein that pokes holes in the outer cell membrane, leaving the inner membrane of the mitochondria intact. It detects differences in cholesterol levels in both cell layers, and it avoids the laborious isolation of mitochondria and lets you use chemicals directly on the mitochondria. So it's a great little advantage to using this PMP. So some of the questions we talked about that you can ask, and you look at cell viability, cell health, cell activation, the different mechanisms, whether it's drug resistance or drug activity, drug effect on the cells. Maybe you're looking at confirming the signaling time point checkpoints, reprogramming, things like that. So many things that you can do with SeaHorse technology. And I'm going to do a time check. We have a few minutes left. And just point you to some resources that you have. And one particular one, which is really nice if you want to look up publications that maybe have the cell type that you're looking at, that you're interested in. Our team has reviewed thousands of publications that mention SeaHorse, and you can pull that publication out with some very interesting information that, again, you can choose the cell type that you want, and you can download some additional information that just save you a little time, where we pull information about the type of instruments they used, the assays that they ran on those particular cell types, and then importantly, how many cells they used approximately. And maybe if you're using a very common cell, then maybe 100 publications, but you can scan quickly through those publications in that Excel downloadable format and see, okay, half of the people use 50,000 cells per well, and the other half used 30. So maybe a good place to start when you're starting out experiments is 40, but then you do a cell density titration to make sure that you get optimal results for your experiment. And again, Anup and others can help you as you get set up. So whether you're using the existing SeaHorse at ASU, or you're going to get ready to use a future one that'll come, just lean on us to help you out. Lots of different webinars that are free and on demand, whether you're looking at immunology, cancer, neurobiology, aging, diabetes, lots of different topics. And then we have monthly trainings, as I've talked about, mentioned many times, but he's available to help you out in any step. So I think that at this point, I will stop and pause for questions, and yeah, open it up for discussion. Let me turn on my video again, make sure. Hi. So okay, any questions?
Yeah. I have another question. It's Miyako here.
Okay, sure.
A couple questions came up. I'm talking with my grad student who's also on the call.
Oh, okay.